My name is Malgorzata Gierynska, and first I would like to thank for inviting me to participate in the third edition of World Congress on Virology and Infectious Diseases. When we look at the history of infectious diseases, only one human disease has been declared to be conquered. I'm talking about one of the most devastating diseases of mankind, smallpox. Smallpox, a highly contagious disease with a high mortality level caused by variola virus, is characterized by a maculopapular rash that converts into vesicles, then ulcers, which become scabs, leaving characteristic deep scars. To combat the disease, the smallpox eradication program was launched in 1966, and in 1980, following the global campaign of surveillance and vaccination, World Health Organization declared that smallpox was eradicated. However, smallpox eradication caused the cessation of the global vaccination program, and now the consequence of this act is the constantly increasing population without immunity to smallpox. Moreover, zoonotic pox viruses are now a growing public health problem, and the recent mpox outbreak can be an example, since vaccination against smallpox with the vaccinia virus provided protection against them, at least partially. Variola virus belongs to the Poxviridae family, orthopox virus genus, together with other pox viruses like cowpox virus, monkeypox virus, and vaccinia virus, as well as animal pox viruses like Ectromelia virus, rabbit pox virus, and several others. Orthopox viruses are large brick-shaped or ovoid virions with double-stranded DNA. Those viruses replicate in the cytoplasm of the target cells and code over 200 genes, including genes encoding protein involved in virus evasion of innate and adaptive immunity. They share some epitopes, and the homology among the surface proteins that are important for attachment and entry to the target cells is very high. This explains the efficient cross-protection elicited by vaccinia virus vaccine. Since variola virus infections are restricted only to humans, and the tight regulations regarding studies with variola virus were set by the World Health Organization, proper alternative animal and cell culture models should be used to recognize immunobiology of orthopox virus infections and understanding the relationship between the host and pox viruses. This knowledge is crucial for developing safer vaccines and targeted antiviral therapies. One of the acknowledged murine models to study the pathogenesis of pox viruses infections is the Ectromelia virus, the causative agent of mousepox, which is a mouse homologue of human smallpox. What is more important, a mousepox model is also used to test medical countermeasures against variola virus and other pox viruses. ECTV is highly infectious to all strains of laboratory mice, but induces lethal disease only in susceptible animals with particular genetic backgrounds and is readily cleared in other strains that are considered resistant. The major difference in the disease is that in susceptible strains of mice during the early stages of ECTV infection, the immune response is polarized toward Th2 profile, while in resistant polarization is directed towards Th1, resulting in the strong induction of cell-mediated immunity. However, it has to be remembered that even resistance can be overcome by the dose and route of infection, as well as it depends on the strains of the virus. The induction of a protective immune response depends on the recognition of infectious agents through the involvement of microbe-associated molecular patterns recognized by membrane-bound or cytoplasmic pattern recognition receptors. Also, the involvement of antigen-presenting cells is crucial. In our studies, using the established cell lines, we focused on the influence of ECTV on the non-canonical NF-kappa B signaling pathway. The final step of this pathway is the formation of RelB p52 dimer that translocates to the nucleus to control the transcription of target genes. Thus, the non-canonical pathway orchestrates, among others, the development of lymphoid organs, humoral immune response, T-cell and dendritic cells function, the progression of inflammatory diseases, and, what is most important, antiviral immunity. Studies done on macrophages revealed that nuclear translocation of RelB was observed in controls stimulated for 4 hours with interferon, gamma, and LPS. ECTV diminished the nuclear content of RelB, but it was not statistically insignificant. However, the nuclear content of p52 was downregulated compared to the control. Further studies indicated that ECTV can counteract the nuclear translocation of RelB p52 dimer induced by different factors. Indeed, in dendritic cells stimulated with PMI ionomycin, ECTV counteracted RelB nuclear accumulation compared to UV inactivated ECTV treatment. The same results were observed in macrophages challenged with ECTV and stimulated for 12, 18, 24 hours with PMA ionomycin, interferon gamma LPS, and LPS respectively. A significant increase in the percentage of nuclear accumulation of RelB was observed. However, RelB was activated in less than 40% of ECTV-infected cells. Moreover, ECTV blocks the phosphorylation of RelB and P100 in macrophages and dendritic cells stimulated with synthetic lipopeptide, PMA ionomycin, and interferon gamma LPS. Also, it prevents the activity of P52. Additionally, 18 hours post-challenge, the analysis of the activity of DNA binding by RelB and P52 in the nuclear fraction of macrophages stimulated either with LPS or interferon gamma LPS and challenged with the virus showed that ECTV actively repressed transcriptional activity of RelB and P52. As I said earlier, the outcome of the mousepox is different in various strains of mice and is controlled by many host immune cells, among which antigen-presenting cells play a crucial part since they are a link between innate and adaptive immunity. Dendritic cells constitute a heterogeneous population of cells in terms of functionality, phenotype, and localization, which is the basis of their classification. It is known that in susceptible strains of mice, the outcome of the disease is characterized by the delayed development of cell-mediated immunity during primary mousepox. However, the role of different subpopulations of dendritic cells in susceptible and resistant to mousepox mice strains was not fully elucidated. Moreover, it was not shown how ECTV affects in vivo those populations. Therefore, BALB/C and C57 Black 6 mice were footpad challenged with ECTV and in indicated time points using magnetic separation, splenic, CDC1 and CDC2 were isolated and evaluated. First, the number of CDC1 and CDC2 were higher in spleens of control C57 Black 6 mice compared to the uninfected BALB/C mice, and a similar relationship was maintained throughout infection. However, at 7 days post-infection, the number of CDC1 and CDC2 in BALB/C mice decreased significantly, but not in C57 Black 6 mice. Both conventional dendritic cell subset were significantly elevated. At this time, severe necrosis was observed in spleens of bulb C mice that influenced the number of conventional dendritic cells. It has to be noted that bulb C mice succumbed to the disease between 9 and 11 days post-infection. The challenge with ECTV promoted maturation of conventional dendritic cells in bulb C and C57 black 6 mice. Surprisingly, the naturally susceptible strains produced highly mature conventional dendritic cells compared to the resistant strain of mice. The evaluation of CDC1 and CDC2 on the 5th day post-infection revealed an increased cell number of both conventional dendritic cell subsets in C57 black 6 mice. Both experimental systems showed the presence of ECTV antigens in conventional dendritic cells despite the strain of mice. The plaque-forming unit test revealed that those cells were actively infected, yielding a higher titer of the virus detected in CDC1 and CDC2 derived from susceptible bulb C mice. Since dendritic cells are essential for the induction of protective adaptive immunity due to the presentation of the epitopes in the context of MHC class I and MHC class II molecules and by the formation of the proper cytokine environment, the profiles of secreted cytokines by both conventional dendritic cell subsets were assessed, as well as their ability to trigger proliferation of CD4 T cells. Among assessed cytokines, interferon gamma was produced at a significantly higher level by CDC1 and CDC2 isolated from C57 black 6 mice starting at 3rd day post-infection compared to cells of bulb C mice. The percentage of IL-12 synthesizing C57 black 6 CD1 increased at the early stages of infection, but the rate of CDC2 producing IL-12 increased at 7 days post-infection in both mice strains compared to the controls. In the case of Th2 profile, a significant increase of IL-4 production was observed only in bulb C CDC1 on 5th days post-infection. ECTV infection significantly reduces the ability of both splenic conventional dendritic cell subset derived from bulb C mice and C57 black 6 mice to induce the proliferation of CD4 T cells compared to the control cells. Moreover, during the early stages of ECTV infection, CDC1 and CDC2 of both mouse strains showed impairment in the triggering of the cytokine secretion by T cells. However, splenic CDC derived from C57 black 6 mice were more effective than those obtained from susceptible mice. Our results showed that ECTV infection in established cell lines of antigen-presenting cells counteracts the activation of P100 and RELB, inhibits the nuclear translocation of RELB, affects the P100-P52 ratio. Moreover, ECTV downregulated the transcriptional activity of RELB and P52, suppressing the formation of pro-inflammatory cytokines. ECTV affects the function of splenic CDC1 and CDC2 derived from susceptible and resistant to mousepox mice strains, revealing the impairment of conventional dendritic cells in both strains. But a higher level of impairment was observed in susceptible bulb C mice. CDC1 and CDC2 obtained from infected C57 black 6 mice produced higher levels of Th1 cytokine profile than conventional dendritic cells isolated from biopsy mice. Nevertheless, conventional dendritic cells derived from infected mice strains presented a diminished ability to stimulate the proliferation of allogeneic CD4 T cells of C3H mice. These data indicate the role of ECTV as a modulator of immune cells. Such a modulation leads to diminished induction of antiviral immunity. This work, and for this I would like to thank my colleagues from our group. This work was done by Professor Lidia Schulz-Dąbrowska, Dr. Justyna Struzik, Dr. Karolina Gregorczyk-Zborow, Professor Felix Stoka, Dr. Matylda Mielcarska, and Zuzanna Biernacka. I have to mention two scientists, Professor Marek Niemiałtowski and Dr. Irma Szporces-Pedes. Those persons were the first scientists who started work with ECTV in our lab. Thank you very much for your attention.